Καλησπέρα σας. Η αναγόρευση του καθηγητή κυρίου Βασιλείου σε επίτιμο διδάκτορα του Τμήματος Χημείας θα ξεκινήσει με την προσφώνηση από τον πρίτανη του Εθνικού και Καποδιστριακού Πανεπιστημίου Αθηνών, καθηγητή μελέτιου Αθανάσιο Δημοπούλου. Welcoming address by the Rector of the National and Capodistrian University of Athens, Professor Meletios Athanasios Dimopoulos.
Κύριε Αντιπριτάνης, κύριοι Κοσμήτορες, κυρίες και κύριοι πρόεδροι, κυρία Ακαδημαϊκέ, αγαπητές και αγαπητοί συνάδελφοι, αγαπητέ κύριε Βασιλείου. Με ιδιαίτερη χαρά, λόγω του σημαντικού έργου της τιμωμένης προσωπικότητας, αποδίδουμε και την παράδοση του Ιδρύματός μας να αποδίδουμε τιμές σε προσωπικότητες που έχουν προσφέρει πολλαπλώς στην επιστήμη. Έχουμε σήμερα την χαρά της απόδοσης της ύψης τιμής ανακήρυξη επίτιμο διδάκτορα του Τμήματος Χημείας της Σχολής Θετικών Επιστημών του Εθνικού και Καποδιστριακού Πανεπιστημίου Αθηνών, του καθηγητή στο Yale School of Public Health, Department of Environmental Health Sciences, κύριο Β.Βασιλείου. Η απονομή του τίτλου αποτελεί ένα φόρο τιμής για την εξαιρετική συμβολή του σε αντικείμενα που αφορούν το περιβάλλον και την υγεία γενικότερα. Ο Βασίλης Βασιλείου είναι καθηγητής και πρόεδρος του Τμήματος Περιβαλλοντικών Επιστημών Υγείας. Έλαβε πτυχίο χημείας το 1983 και διδακτορικό στη βιοχημική φαρμακολογία το 1988 από το Πανεπιστήμιο Ιωαννίνων. Στη συνέχεια εξειδικεύτηκε σε θέματα αλληλεπιδράσεων γονιδίου και περιβάλλοντος, στην μοριακή τοξικολογία και στην φάρμακογενετική στο τμήμα περιβαλλοντικής υγείας της Ιατρικής Σχολής του Πανεπιστημίου Σινσινάτι. Το 1996 εντάχθηκε στο Πανεπιστήμιο του Κολοράντο, όπου διήλθε όλες τις βαθμίδες για να εξελιχθεί σε καθηγητή και διευθυντή του μεταπτυχιακού προγράμματος τοξικολογίας. Από το 2008 ήταν επίσης καθηγητής οφθαλμολογίας στην Ιατρική Σχολή του Πανεπιστημίου Κολοράντο. Τον Ιούλιο του 2014 εντάχθηκε στο δυναμικό του Πανεπιστημίου του Yale. Ο καθηγητής Βασιλείου έχει καθιερωθεί διεθνώς και το ερευνητικό του έργο την χάνει ευρύτερης αναγνώρισης. Τα ερευνητικά του ενδιαφέροντα περιλαμβάνουν την αιτιολογία και τους μοριακούς μηχανισμούς της ανθρώπινης νόσου που προκαλείται από το περιβάλλον, όπως η υπατική νόσος, η παχυσαρκία και ο διαβήτης, ο καρκίνος και νευροεκφυλιστικές παθήσεις. Η έρευνά του επικεντρώνεται στα μέσα με τα οποία το exposome, το σύνολο δηλαδή των παραγόντων που ο άνθρωπος εκτίθεται, ο μεταβολισμός και τα αντιοξυδωτικά συμβάλλουν στην ανθρώπινη υγεία και την ασθένεια. Το εργαστήριό του χρησιμοποιεί υπερσύγχρονες ολοκληρωμένες προσεγγίσεις και μεθόδους με διεθνή αναγνώριση. Ο καθηγητής Βασιλείου έχει δημοσιεύσει πάνω από 250 εργασίες με υψηλότατη απήχηση διεθνώς και έχει επιμεληθεί 3 βιβλία. Μετέχει σε συντακτικές επιτροπές πολλών περιοδικών και σε επιστημονικά συμβούλια. Έχει επιβλέψει 16 διδακτορικές διατριβές, 15 μεταδιδακτορικές έρευνες και 25 διπλωματικές εργασίες. Για το έργο του και τη συνολική προσφορά του, την πλούσια συγγραφική του δράση και την αξιόλογη ερευνητική του δραστηριότητα έχει λάβει σημαντικές διακρίσεις και πλήθος βραβεύσεων. Για όλα τα παραπάνω, είναι ιδιαίτερη χαρά και τιμή να υποδεχθούμε τον κύριο Βασίλη Βασιλείου ως επίλεκτο μέλος, ως επίτιμο διδάκτορα του Τμήματος Χημείας του Πανεπιστημίου μας. Τον ευχαριστούμε για την σημαντική προσφορά του στο περιβάλλον και στην υγεία γενικότερα και βεβαίως τον ευχαριστούμε για τη συντέλεση σημαντικά στη δημιουργία αυτού του νέου κέντρου μεταξύ του Πανεπιστημίου Yale και του Πανεπιστημίου μας που αφορά στο περιβάλλον, στην υγεία και στις συνέργειες διαφόρων τμήματων και σχολών των 2 πανεπιστημίων. Σας ευχαριστώ πολύ.
Παρουσίαση του έργου και της προσωπικότητας του τιμωμένου από τον καθηγητή του Τμήματος Χημείας της Σχολής Θετικών Επιστημών του Εθνικού και Καποδιστριακού Πανεπιστημίου Αθηνών και αναπληρωτή πρόεδρο του Τμήματος Χημείας Νικόλαο Θωμαίδη. Presentation of the distinguished career and accomplishments of Professor Β.Βασιλείου by Professor of Chemistry Νικόλαος Θωμαίδης.
Αξιότιμοι κύριε Πριτάνη, Αντιπριτάνης, Κοσμήτωρες Πρόεδροι, dear distinguished guests, dear Professor Βασιλείου, dear Βασίλη. It's a great honor and a deep personal pleasure for me today to present the work of Professor Βασιλείου on the occasion of his nomination as an honorary doctor of the Department of Chemistry of the National Capodistrian University of Athens. Professor Βασιλείου is most certainly a recognized and acclaimed scientist in the compelling and innovative fields of molecular toxicology, pharmacogenetics, genomics, environmental health sciences, gene-environment interactions, and exposomics. Nonetheless, he is a very dear friend and a great collaborator. The academic career of Professor Βασιλείου is indicative of his scientific consistency, constant evolution, and his continuous devotion to fascinating, innovative, and influential aspects of his field. Starting his career at the University of Ioannina, following his bachelor in chemistry, he focused on life sciences, obtaining a PhD in biochemistry, followed by a postdoctoral fellowship in pharmacology. Even in these early stages of his career, he very much valued, having already recognized the great importance of interdisciplinary research and the connection of the scientific research to the improvement of public health and well-being. The United States of America and the University of Cincinnati was Vasilis' next stop. His research there expanded in the fields of molecular biology, toxicology, and pharmacogenetics, opening new spectacular horizons. Moving westward in the States, he was appointed in 1996 as an assistant professor at the Department of Pharmaceutical Sciences of the School of Pharmacy at the University of Colorado Health Sciences Center. There, during his career as a director of the Molecular Toxicology and Environmental Health Sciences program, been appointed as an associate professor in 2001 and a full professor in 2007, worked on the impact of alcohol and its metabolism to cancer and the discovery of early biomarkers of certain types of cancer. These are few examples of his pioneering and outstanding work there. Since 2014, Professor Βασιλείου is serving as a professor at Yale University. He is affiliated with the Department of Ophthalmology and Visual Sciences in Yale Medical School, with Yale School of the Environment, and with the Department of Environmental Health Sciences in Yale School of Public Health. His outstanding work has rightly secured substantial funding both for supporting new trainees as well as for forging and reinforcing collaborations and powerful research network as those that we are all involved and we are forming now in the University of Athens. His research activity is indeed very impressive in numbers, with more than 250 publications in some of the most renowned journals in his field, more than 17,000 citations, an age index of 65, according to Google Scholar database. Over and above that, if I might say, it is even more impressive quality-wise, as his research efforts have led to more and some fundamental results and conclusions, and have produced and developed pioneering approaches that open new paths both in terms of methodology as well as in a wide range of applications. His books on the relation of alcohol and cancer are referential on the field and used worldwide. He is also editor-in-chief in Human Genomics Journal, while he serves as an associate editor or member of the editorial board in 5 other scientific journals covering a broad range of topics. Β.Βασιλείου and his valuable contribution to research and teaching has been internationally recognized, receiving numerous awards and honors. Mentioning some of the teaching recognition Professor Βασιλείου has been awarded, such as the Teacher of the Year Award, selected by the Pharmacy First Professional Year class in 2001, and the Dean's Mentoring Award in the University of Colorado. It would be an unforgivable omission not to praise his devotion to his students, his inspirational lectures, and his passion in supporting and training young scientists. He has supervised 16 doctoral theses, 5 more are ongoing at the moment, 15 postdoctoral researchers, and more than 25 graduate theses. His enthusiasm in serving sciences, inventing new ways of thinking, and in training future generations of scientists is exemplary. During his long career, international recognition has not prevented him from always keeping very strong connections with Greece, welcoming Greek students, and supporting scientific collaborations. To conclude, it is a great honor and pleasure for me personally, but I'm sure I also speak on behalf of all the members of our department, to welcome Dr. Vasilis Vasiliou to the Honorary Doctors of the Chemistry Department of the National and Kapodistrian University of Athens. We are looking forward to a firm, creative, and fruitful collaboration long term. Dear Vasili, thank you very much, and please accept our most sincere welcome and congratulations. Thank you very much. Θέλουμε να καλωσορίσουμε με την ευκαιρία αυτή την Υπουργό, αναπληρωτή Υπουργό Υγείας, την κυρία Κάκα, και να πούμε ένα μεγάλο ευχαριστώ που είστε σήμερα εδώ, εκπροσωπώντας και το γραφείο του Πρωθυπουργού, τον ίδιο τον Πρωθυπουργό. Ευχαριστούμε πάρα πολύ. Επίσης, να καλωσορίσω και την Γενική Γραμματέα, τη Χριστιανά την Καλογύρου, η οποία μας τιμά επίσης με την παρουσία της. Ευχαριστούμε πάρα πολύ που είσαστε εδώ.
Αναγόρευση του τιμωμένου. Ανάγνωση των κειμένων του ψηφίσματος του τμήματος, της αναγόρευσης και του διδακτορικού διπλώματος από την πρόεδρο του τμήματος, καθηγήτρια Χριστίνα Άννα Μητσοπούλου. Conferring ceremony. Reading of the chemistry department's citation and conferring documents and of the diploma by the chair of the department, Professor Christina Anna Mitsopoulou.
Εθνικών και Καποδιστριακών Πανεπιστήμιων Αθηνών. Ψήφισμα του Τμήματος Χημείας της των Θετικών Επιστημών Σχολής. Τύχη αγαθή. Πριτανεύοντος εν το Αθήνης Εθνικό και Καποδιστριακό Πανεπιστημίο Μελετίου Αθανασίου Κ. Δημοπούλου, κοσμητεύοντος εν των Θετικών Επιστημών Σχολή Ιωάννου Π. Εμμανουήλ και προεδρευούσης εν το Τμήματι Χημείας Χριστίνης Άννας Δ. Μητσοπούλου, έδοξε νομοφόνο στο Τμήματι Βασιλείων Κ. Βασιλείου, άνδρα έφετε πεφεκότα και παιδείας έμπλεων, εξ Ελλάδας ορμημόμηνων και εν κόσμο των νέων εγκατεστημένων, ένθετες περί την χημίαν και δι και την βιοχημικήν φαρμακολογίαν και την φαρμακογενετικήν και το καλούμενον γονιδίωμα σπουδάς μάλα προαγαγόντα και αρτιώσαντα, πολλές και καινοτόμεις συγγραφές, εν της ομοτέχνης διαλάμψαντα. Παμπόλους μαθητάς δε διχώτα, Ινστιτούτων Ελέας και Υγείας, εν τη πέρα του Μεγάλου Ωκεανού γης ειστασάμενον, έγλην ζηλωτήν κτισάμενον και επένων αγλαών αξιοθέντα. Προς δε τούτη σέβνουν την πατρίδα, αίδια τελούντα και το ημετέρω τμήματι, εν επιστήμη παραστάτην δεξιών αεί γνωριζόμενων, έδοξεν λέγω. Αυτόν τε ένεσε και επίτιμον διδάκτορα του τμήματος αναδείξε, το δε ψήφισμα τόδε, μεμβράνη αναγράψε, και την του τμήματος πρόεδρον δημοσία εν τη αιθούση τη μεγάλη αναγνούσαν και τα στιμάσαν υπούσαν, επιδούνε αυτό. Ή αν ημέρα η αναγόρευσης γέννηται, καθάν ενόμιστε. Εγένετο, Αθήνηση, μηνός Σεπτεμβρίου, Τετάρτη και 20, έτους πρώτου και 20 και δυσχιλιοστού από Χριστού γεννήσεως. Η πρόεδρος του Τμήματος Χημείας, Χριστίνα Άννα Δέλτα Μητσοπούλου. Αναγόρευσης. Επιδίπερ το Τμήμα Χημείας της των Θετικών Επιστημών Σχολής του Αθήνησιν Εθνικού και Καποδιστριακού Πανεπιστημίου Βασίλειον Β.Βασιλείου άξιον απέφυνε του επίτιμου διδακτορικού αξιώματος η δε των Θετικών Επιστημών Σχολή τούτα παιδέξατο και ο πρίτανης της σχολής γνώμη επινεύει διατάφτα εγώ, Χριστίνα Άννα Δέλτα Μητσοπούλου, καθηγήτρια της Ανοργάνου Χημείας, νυν δε του Τμήματος Πρόεδρος Χημείας, χρώμενη τη δυνάμει, ειν παρά το Πανεπιστημιακών Νόμων και του Τμήματος έχω λαβούσα, Βασίλειον Β.Βασιλείου, επίτιμον διδάκτορα του Τμήματος Χημείας της των Θετικών Επιστημών Σχολής, του Αθήνησιν Εθνικού και Καποδιστριακού Πανεπιστημίου, δημοσία αναγορεύω και πάσα σε αυτό τας προνομίας, τα στο πανεπιστημιακό τούτο αξιώματι παρεπομένας απονέμω. Εγένετο, Αθήνησιν, μηνός Μαΐου 10, έτους Δευτέρου και 20 και δυσχιλιοστού από Χριστού γεννήσεως, η πρόεδρος του Τμήματος Χημείας, Χριστίνα Άννα Δέλτα Μητσοπούλου. Ελληνική Δημοκρατία. Πριτανεύοντος εν την Αθήνηση, Εθνικό και Καποδιστριακό Πανεπιστημίο, μελετείου Αθανασίου Κ. Δημοπούλου, καθηγητού της Θεραπευτικής Ογκολογίας Αιματολογίας, κοσμητεύοντας εν των Θετικών Επιστημών Σχολή, Ιωάννου Π. Εμμανουήλ, καθηγητού της Άλγευρας, προεδρευούσης εν το Τμήμα τη Χημείας, Χριστίνα Άννης Δέλτα Μητσοπούλου, καθηγήτριας της Ανοργάνου Χημείας, Βασίλειον Κ. Βασιλείου, αποδόγματος ομοθήμου, της των Θετικών Επιστημών Σχολής, εις τους επίτιμους διδάκτορας εγκρίνομεν, Αθήνηση, μηνός Μαΐου 10, έτους Δευτέρου και 20 και δυσχιλιοστού από Χριστού γεννήσεως. Ο πρίτανης, μελέτιος Αθανάσιος Κ. Δημόπουλος, ο κοσμήτορ Ιωάννης Π. Εμμανουήλ, η πρόεδρος του Τμήματος Χημείας, Χριστίνα Άννα Δέλτα Μητσοπούλου. Συγχαρητήρια.
Περιένδυση του τιμωμένου με την τίβαινα της σχολής από τον κοσμήτορα της Σχολής Θετικών Επιστημών του Εθνικού και Καποδιστριακού Πανεπιστημίου Αθηνών, Ιωάννη Εμμανουήλ, Investiture of Professor Βασιλείου with the gown of the School of Science by the Dean, Professor Ιωάννης Εμμανουήλ. Ομιλία του τιμωμένου καθηγητή Β.Βασιλείου. Speech of Professor Βασίλειος Βασιλείου.
Okay. Perfect. Αξιότιμη κυρία Υπουργός, αξιότιμη κυρία Γενική Γραμματέας, αξιότιμε κύριε Πρίτανη, αξιότιμοι κύριοι Κοσμήτωρες, αξιότιμη κυρία Πρόεδρος του Τμήματος. Να το πω λίγο στα ελληνικά. Τι τιμή είναι αυτή που I'm starting to get emotional, especially for such an honor to be here and to be honored the PhD degree. From the bottom of my heart, I really thank you very much, and I wish my father was here. Anyway, I'm sorry for being emotional, but I'm Greek. So, thank you very much for all your good words, and I'm gonna start a little bit with a combination of how I started, what I have been, and what we're doing right now. Nikos mentioned I have a passion for education, and this is true. I like to really educate not only students, but also mentor junior faculty. And this couple of slides will tell you how I started my work at the University of Ioannina with my advisor, Marios Marcellos, who introduced me to the field of the alcohol metabolism. And then we moved to aldehyde dehydrogenases on the left, reactive oxygen species, and liver disease. From there, I also start by leaving his laboratory and going to University of Cincinnati. I had a feeling that glutathione would be a major player in this whole pathway. So then I went to Yale, and all through the Cincinnati and Yale, I had this passion about the emerging contaminants, and especially water contaminants, and how this can affect cancer and liver toxicity, and how they would be the interplay. In the studying of interplay between aldehyde dehydrogenase and glutathione, we found significant roles for glutathione in the eye development, diabetes, immune system, and lately we're going to the neurobehavioral effects and the effects of the exposome. So what I have in the bottom of this slide are the four major places in here, which I started with University of Ioannina. Then I moved to University of Cincinnati. With that, I have to tell you that I was awarded a Fogarty Fellowship to go to Dr. Sekelaridis in University of Indianapolis, and I want to thank him from the bottom of my heart because he was one of the great mentors that I had during my early career. Then, from Cincinnati, I got my first appointment, as you heard, in University of Colorado, School of Pharmacy. And I wasn't planning of leaving Colorado. Colorado is beautiful. I had bought a beautiful house, but Yale came after me, and who's going to say no to Yale, especially when they offer you a chairman position for the environmental health sciences? So here I am, and here is I am at Yale. So what I'm going to do today, I'm going to try to speed up a little bit because I have a lot of things to cover. I don't cover all the areas, but I have chosen a few things that it will show my personality and my work. So we're going to start with defining the exposome and going to the emerging contaminants. And I'm going to talk to you about the 1,4-dioxane and the co-occurring contaminants, which we just recently got funded with my colleagues here from Yale. And then I'm going to go to per and polyfluorinated alkyl substances, the forever chemicals PFAS. I'm going to move a little bit to the alcoholic liver disease. I'm going to show you what the power of new technologies are and where they are taking us. Then I'm going to talk to you, my basic lab given or introduced by Myros Marcellus was aldehyde dehydrogenases. And then we moved to glutathione, and glutathione redox biology is a baby that has given us a lot of surprises in our process to getting all these discoveries. Lately, we are paying a lot of attention on the exposome and neurodevelopment and neurodevelopmental disease. And finally, I'm going to close with a reflection of what I have done. As I told you, I went to Yale, and my Department of Environmental Health Sciences has a mission to advance scientific knowledge about the connections between environment and health through research and education, the next generation. So one of the things that I did once I went to Yale is I wanted to have an illustration of what my new department would be and what we will be doing. And this picture, it tells you all the exposures that we are being exposed every day. And that includes not only the chimneys from the industry, not only the polluted waters that we have, but also some other factors, which is diet, lifestyle, of course smoking and drinking, but also stress. And by the way, Dr. Ko, I designed that before COVID-19, before SARS-2, and you can see that we had the viruses in there, which I believe is a part of the environment and the interaction between the environment and the viruses and the microbial diseases in general. So the whole idea is we are interested in total exposures. We are exposed to millions of compounds on a daily basis, okay? To thousands, I would say, well, not millions. So what is the exposome? Chris Wilds, in 2006, developed this concept of exposome. And what is exposome? Is the totality of exposures from preconception, not conception, preconception to death. So what we have been exposed, even our fathers or mothers, and then how this has affected our lives. And this is what we call as a total exposures. One of the challenges, though, is how do you determine that? How you look at the effects of this, all these chemicals to the health outcomes. So there are a lot of areas that you can do. You can measure as much as you can as total exposures. You can take different times. You can measure with the advanced technology, as we will say, the mass spectrometry. You can determine the chemicals present in your body. You can also see changes into your metabolome that will reflect to what chemicals you have been exposed to. And then you can use the artificial intelligence to incorporate all this to get modeling systems of the exposures and associations to human health. So, as I said, exposome was in my heart as long as I went to this department. So immediately I organized a Yale symposium on lifetime exposures and human health. Dean Jones was there. Dean Jones is one of the fathers of the exposome from Emory University, and we have the National Institutes of Environmental Health Sciences present there. And we develop all these concepts and set up a framework essentially for my department to move ahead with that. And that was the basis that I tried to do all the recruitments in the department that we can devote, from exposure assessment to all the changes that they may occur. We have a very good publication that it can be found on that, and this was essentially the beginning of our journey to the exposome analysis. So again, this is the illustration of our department, and one of the things that I did once I went to Yale is I wanted to be in touch with local authorities. So when I'm saying local authorities, the state agencies. So I went to the Department of Public Health and to the Department of Energy and Environmental Protection, and I sat down on a table with 30 people, and I said, I would appreciate if you tell me what is the need of the state of Connecticut that we need to study. What is the chemical that you need to study? And that time, PFAS have not surfaced yet as emerging contaminants. And what they all told me, 1.4 dioxane and water contaminants. So we did focus on the water contaminants. And emerging water contaminants is a term which essentially is very new. So what is an emerging contaminant? Are chemicals that they are detected in public water supplies, on water supplies in general, throughout the globe, that is not recognized as a public threat. We don't know anything about, in terms of epidemiology, we don't know anything about the mode of action, and we don't know how does it occur. And those include the chemicals that include pharmaceuticals, personal care products, pesticides, herbicides, industrial chemicals, and of course, endocrine disrupting compounds. Where they can come from, you can see from the picture, they can come from the sewage, from the runoff, everywhere, and they can end up to the drinking water. And the problem with the chemical that I'm going to talk to you about, which is the 1.4 dioxane, is the state agency said, this is a chemical that has been used as a stabilizer. We cannot filter it. There is no way you can filter it. It passes throughout the system, and people is drinking it on a daily basis. So can you do something about it? And I said, I think we can do. Anyway, to make a long story short and show you how important it is, it has been also named as another forever chemical. And recently, if you can see from the ECHA, which is the European Chemical Association, if I remember well, they just announced that this chemical is of potential significance, or potential threat for European population as well. So it's not only in the United States, it's everywhere. It's a stabilizer, and this is the 1.4 dioxane. It's found everywhere. This report, Dr. Anastas sent me that link a couple of months ago, indicating that they found in the public water of New Jersey, they found this chemical. And the problem with that all through this year is nobody was thinking about the toxicity of this chemical. Nobody was thinking that they would measure this chemical into the drinking water. And it is possible that we're drinking it on a daily basis. So what do we do with that? So immediately, I wanted to create, and I don't know about the quality of the slides, I created a group of people. So I tried to bring together the School of the Environment, the Department of Chemical and Environmental Engineering, to work together with us on environmental science projects and biomedical projects to develop to gain knowledge about the 1.4 dioxane in terms of remediation, in terms of exposure assessment, and determining the mechanism of function of this particular chemical, which, by the way, I forgot to mention, it is characterized by International Agency for Research on Cancer, and also by the Environmental Protection Agency and the National Toxicology Program as an animal liver carcinogen. In other words, you put it in the drinking water, the mice will develop cancers. And liver cancer was one of the major outcomes. So after working really hard with my colleagues, quite a few of them are today here with us, we worked really hard and we put a center together. I'm really lucky to have such a great colleagues and to lead that center of, it is the NIH Yale Superfund Center. It's been about less than a month that we get the final word that we're getting money from the National Institute of Health. And this is a multi-center consisting of 4 cores, including an administrative core, a community engagement core, and also the training core. And also it has the administrative core where Jae-Hong Kim, which is here, is my deputy director, and Paul Anastas, which, by the way, was a White House advisor on environmental issues, White House, and several presidents, if I remember well. He is our research translation coordinator. So this center, in addition to the course and the training that we have 2 projects on biomedical research projects, and we have 2 environmental sciences project. I am so proud and so lucky to be with colleagues like that. We have members of National Academy of Sciences, which are included in this center. And I'm not going to name, but our team, I'm so lucky again to be with these people and work with that. So what I'm going to do is, I know it's very difficult to read everything from there. I'm going to give you just a taste of what we're doing with one of the project of the Project One, which is my lab essentially, along with one of my trainees, which now she's going to become a completely independent scientist. And this is the mechanistic studies of 1.4 dioxane for liver toxicity and carcinogenesis. Remember what I said, emerging contaminants, we don't know what the mode of the action is. We know that they can cause cancer, but we don't know how. So this project, it involves transgenic knockout mouse models and also zebrafish models that we study not only the 1.4 dioxane, but also its co-occurrent contaminants that they can be found in the soil and in the drinking water that we have. And we're using multi-omics system approach, which consists of transcriptomics, metabolomics, and proteomics, and then we're doing the phenotyping on that in terms of the health outcomes. Then for this project, we're utilizing artificial intelligence and deep learning to combine all these effects and trying to make the mode of action mechanism. We have actually done quite a few of the studies in here. What you see, and very briefly, it's dioxin on the left, and this is the 3 metabolic pathways that it can go to the final product, which is an acid that we can determine, and we can find that, we can measure it with the mass spectrometry. One of the points I want to mention here is you can see that this chemical is metabolized by cytochrome P452E1. I don't want to make you tired. I want to give you the big picture in here. What we have identified, the first 2 papers, is we found that this chemical can interact with your DNA, and it can also inhibit the DNA repair mechanisms. This is the 2, I think, monumental papers in terms of the mechanism that my laboratory has found and has contributed to the research. Well, to make life a little bit more complicated, this is, I'm not going to spend too much time in here. This is what we believe it occurs with the study so far, but we're doing more experiments to define the mode of actions as a liver carcinogen. One of the things I want to bring your attention to that is it has the same mechanism by which probably alcohol induces liver cancer as well. So imagine if you're talking about alcohol and you're drinking every day water that has 1.4 dioxane, you can still have some combinatory effects. So this is, again, it's a little bit difficult mechanisms to do that. So I'm going to give you a lighter taste here, since we have Jae-Hong Kim in here. He is the leader of our Project 4, which is, okay, you have a chemical, you have an emerging contaminant. How do you get rid of it? Okay, how you can provide clean water for your people. So what Jae-Hong does is trying to break down, as I told you, 1.4 dioxane cannot be filtered. It goes through the filters, whatever. You try everything you can, 1.4 dioxane it goes. So here is Jae-Hong on the left. He is also there. He was touring the Acropolis yesterday, but in addition to be a tourist, his laboratory has developed this electrocatalytic advanced oxidation system that it can break down dioxane. Okay, at this time it might be a little bit expensive, but he's trying to make devices that can be available to housing and, you know, really purify the water on that. So this is the advanced oxidation used for 1.4 dioxane, and, you know, he has started using that for perfluorinated compounds, PFOA, at this time. Again, I'm not going to give you great details on this, but if you want more, you can discuss with him after this talk. So, but this is a very good transition slide to show you that we have the PFAS substances. And these PFAS substances, actually, I was really surprised. Last week, I gave a talk to the medical genetics in University of Rome. So I had an audience of about 120 to 130 students and professors. So I asked them, does anybody know what PFAS are? And the answer was no. And the answer was no for most of us about 5 or 6 years ago, because this had never been coming as a public threat. This is what's happening the last 6 years. So this is two models. And essentially it has been done, DuPont and the 3M, they developed these molecules, and I will show you a little bit how they develop it. But essentially, they replace the hydrogen atoms with fluorine atoms into these carbon structures. This bond, the carbon-fluorine bond, it's impossible to break. And this is the major problem, and this is why it's called forever chemicals. So they're widespread everywhere. The seating that you have in your chairs right now, most of the clothing, and I will show you a few more, we have them on a daily. If we measure, if I take your bloods right now, I can measure easily 50 or 60 of those PFAS in our blood. This is the truth. I'm not trying to make, threat to give you a fear, but this is the reality. And we didn't know anything. So they're widespread. There are numbers of exposures, but the most concerning thing is they stay in the ecosystem, they cannot be broken down, and once they get into the body, it's very difficult to eliminate them. Okay, you cannot, and they stay forever. So as I said, they started very early, 1930s. They found these chemicals and they started utilizing them. Initially, they put them in the atomic bombs to protect them against the corrosive, and they actually initially was synthesized to cover the tanks, so the tanks could be protected from the rain and everything. So then they decided in the 90s, they found that this Teflon, this PFAS, they're non-stick. So they put it into the pans, and the non-stick. So if you have, my personal opinion, if you have non-stick pans, throw them away immediately. That's at least we did. So after the non-stick pans, they went to the foam that they used to stop the fire. So this is another major public threat. And then from there, after this exposure, it went widespread. Every plastic that you can have, it has the PFAS. And now, the evolution of that is the industry. How many they have been withdrawn? Only 5. Not withdrawn from the company, not from the 3M. Why? Because there were a number of lawsuits, which I will tell you how it happens. EPA has not been taking, which is the Environmental Protection Agency, has not taken any measure to eliminate those from there. So there is a major thing. How many they are? We started with a few. Right now, there are about 9,000 chemicals of those PFAS available that the industry makes for all these products. And it's very scary. Where you can find it? You can find it, as I told you, everywhere. From your jackets to protect the rain, the pants, the plastic, your tires, the food packaging. The food packaging, which is very scary. The food that you're taking, your pizza, and because of that, it can have the PFAS which are transferred to your food. So this is how they can be distributed from the manufacturer, and this is what it can end up to your water, going through as a fugitive or to the aqueous waste system. It can go to the cleaning systems, and then it can be as a sludge into your agricultural systems. It can be used to the occupational exposures, consumer reports, consumer uses, and so on. But what I'm going to say is they exist everywhere, and even if they go into the air that you can breathe them, but the rain precipitates them back. You burn them, they're going to go up, they're going to come back down again. They're there, they're over there. And what the adverse effects of the PFAS, they cause certain, now, even within 5 years, and with 10 years of research, they have been characterized as causing cancer to the kidney and to the testis, and they are also causing some liver toxicity and so on. And we still find out more of the effects of the PFAS, and we're doing a lot of studies on that. One of the things I want to tell you is that the PFAS are probably responsible also for the interaction with our immune system, making us more sensitive to COVID infection and the adverse effects of the COVID, of the infection and the idea of health outcomes. So, along with Linda Hadji's group, who is another Greek in University of Southern California, we have done a lot of studies. This is systematic review. We found out both animal and human studies. It's the first study that shows both in animals and humans that the PFAS cause liver toxicity, period. And this was the first study. The National Institute of Health was very excited about our study, and they invited a perspective to show how important was our study in terms of the PFAS, in terms of liver toxicity. So, talking about that, this is a title that was actually given from Paul Anastas in terms of challenges and opportunities. Some people see challenges. It's a challenge to have the PFAS, but others seeing as an opportunity to do more research. So I organized another Yale symposium of the PFAS that we tried to bring both local authorities, state agencies, community people to talk about the PFAS. And that's where, how you can, utilize your green chemistry, how you can utilize your analytics, how you can utilize the mitigation, remediation of the PFAS, and essentially how you can be in touch with the people and notify that. In addition to that, I have also done town hall meetings. Even in the COVID, during the COVID, we hold them as an online. And if you have not seen the movie, and if you have the Netflix, I really beg you to watch this movie. This movie called Dark Waters, and it tells you the whole story, how the PFAS have surfaced the area of research and how, they have been the lawsuits. And this was due to a lawyer, an environmental lawyer named Robert Below, which actually I have him as a lecturer in my department, and he's a fantastic guy. But I really want you to see the movie because you can see the entire, how it started, how the PFAS went to the cows, to the people, how they can cause cancer to the people. It is an amazing movie, and there is a book also called Exposure. So we hold these town hall meetings, and in that town hall meetings we had state agencies, we had Robert Below, of course, and we had Linda Berbam, which was the former director of the National Institutes of Health. So, as you can see, is the exposome is total exposures that you get in. It's still in its infancy, and there will be exciting discoveries in the future. So the emerging contaminants are a significant public health issue, and the more we learn about them, the better we will prepare to protect the general population. Very quick transition slide. One of my students is looking now on the PFAS. Remember, I told you PFAS is causing liver toxicity and liver, it might cause liver cancer. But one of my ideas was, can this interact with alcohol and potentiate the liver toxicity of the alcohol? So we are doing so far using the enhanced cohort. We don't see any interaction because we don't have enough data, but we're doing more. But this is, again, a very good transition to show you that I have been working with the alcoholic liver disease, which, alcohol abuse takes normal liver, goes to steatosis and then to fibrosis, and a small percentage will go to cirrhosis, and then finally will go to hepatocellular carcinoma. It is well-known described effects, and of course you can have also the alcoholic hepatitis, which is a life-threatening disease due to the alcohol. So, as I told you at the beginning, I started going to the oxidative pathways of alcohol metabolism because alcohol induces toxicity through these mechanisms. What I have for you in here is my first ever published paper from Marios Marcello's lab on agents that they can cause alcohol intolerance phenotype. In other words, if you take the medication and you take the drug, you take alcohol, you're like drug. This is what the physicians may know as a disulfiram-like reaction, and this is essentially the way that disulfiram has been developed as an anti-abuse to protect alcoholism. Very quick again, don't want to take too much of your attention, is ethanol is metabolized to acetaldehyde, and then it goes to acetate, and then to acetyl coenzyme A. But molecules in here is acetaldehyde, which can create DNA and protein adducts, and also the metabolism through the cytochrome P450s, which generates reactive oxygen species, which can cause glutathione depletion. Now, this acetaldehyde, if it doesn't get metabolized, it can go as, again, to reactive oxygen species and can generate more lipid peroxidation, more aldehydes, and you can keep going and going, and as a method of toxicity. So this was a very big mechanism that I've been studying for all these years. I have knockout models and everything, which I'll show you in my next slide. Very quick. This is the center that I have created, and it's funded from the NIAAA, which is the National Institute for Alcohol Abuse and Alcoholism. And essentially what I have developed is a crosstalk between alcohol metabolism and also glutathione, which is your major antioxidant. And the major what you see in your with your red lights are the enzymes that we have generated transgenic knockout models. In other words, we take the mouse and utilizing genetic mechanism, we delete that gene completely. And we create models to study the role of these genes into the mechanism. So we have created this NIAAA. It's in its 10th year of funding from there, and we're studying the mechanism. But I didn't stop in there. I'm always eager to learn more and do that. So once I moved to Yale, I have used my startup, and I got really high-end analytical mass spectrometers to use, as Nico said before, for mass spectrometry and metabolomics. So this is some of the papers we just recently published on lipidomics, redox lipidomics, and metabolomics on early changes into the liver. We can identify changes into your plasma, into your liver, much far in advance before any pathology can be shown. Any pathology. And this is the whole idea. How we can develop really early biomarkers, say, you know what, you have to stop drinking immediately. And this is before you start seeing even the fatty liver. This is truly amazing, and it's going through. Just to show you, this is also the tissue imaging. So you take the piece of the tissue, you're running it through the laser, you acquire the chromatographs, and then you can do a special distribution of your molecules, lipids, into the liver, and then you can identify what is exactly the special mechanisms of toxicity. And as I said, this was the initial data that we have generated, and then now this is with a new machine that both my lab and Nico's lab are actually acquiring at the same time, which is a timsTOF from Bruker, and the sensitivity and the accuracy of this is simply amazing. This is how the technology is advancing, and you can see really very high resolution on your studies, which is truly amazing. I'm going to start. I know I'm get you tired, but I'm going to give you some of the examples of our glutathione, which for me are the most fascinating research that I've performed in my life. And as I said, glutathione is your major antioxidant. And so essentially glutathione is a tripeptide, 3 amino acids. It's glutamate, cysteine, and glycine. So when you bring the two first amino acids together, there is an enzyme called glutamate cysteine ligase, which is the rate-limiting enzyme. In other words, is the enzyme which controls the entire biosynthesis of glutathione. And this enzyme is a whole enzyme. It's a complex enzyme consisting of 2 different proteins, 2 different genes, the GCLC and the GCLM. GCLC is the catalytic subunit, GCLM is the modifier subunit. So if you lock the gene of the catalysis, there is no life. Your mice will not survive, they will die before even they get born. And this is how critical glutathione is for life. But if you knock out the gene of the modifier, you get mice that are perfectly fine. They only have less glutathione. So what was a perfect model system to start the alcohol metabolism and everything in there? So just very briefly, when you knock out glutathione in liver disease, when you knock out specifically into the liver, you knock out your glutathione synthesis into the hepatocytes, eventually you're losing your animals from acute liver failure, which is associated also with cirrhosis. And we rescue this phenotype by adding glutathione precursor into their drinking water, so replenishing the loss of glutathione. They survive the acute toxicity, but they still develop cirrhosis. This indicates how vital is the role of glutathione in the liver disease. On the other hand, when you knock out the modifier unit, which brings the glutathione level in the 15% level, the mice are perfectly fine. And actually, to our surprise, these mice were completely protected. When we put them on alcohol, because they had lower levels of glutathione, I was expecting that the mice will have a severe phenotype on alcohol. On the contrary, and to our surprise, these mice were completely protected. So imagine you have your defense system at the 15% compared to the other livers, and the mice are perfectly fine. And not are perfectly fine for alcohol toxicity, they're perfectly fine in insulin resistance. These mice will never develop any diabetes or any obesity if you put them on high-fat diet, which is the paradigm. We could not believe the results that we found. At the beginning, we had to repeat 3 times the experiment because we thought we were mixing the samples. And you're talking for big animals. So anyway, to make a long story short, and I don't want to make you tired again, what we found is there is an oxidative stress in these mice. Because they have lower levels of glutathione, they have a cellular response to this change. And this is what it's called hormesis. Hormesis is a response of your body to oxidative stress. Oxidative stress is not bad in all the cases. So in this particular case, what happens is the oxidative stress increases the AMPK, which is the master regulator of metabolism, and these mice are completely protected. Fatty acid synthesis does not occur. Fatty acid oxidation is happening. So, of course, many people say, can we do that to protect from alcohol drinking? But that's not our intention. Anyway, to make a long story short, very quick, we have done a lot of metabolomics. We have really identified everything. One of the things that I'm going to say that I'm proud, I'm not going to go to the right scheme, is Dr. Chen, which is the first author on these 3 papers. This has been with me training, and she just got her first R01, which is it's a $2 million grant that she got on her own. I have nothing to do with that. This is a graduate student who came to my lab as a postdoc and evolved down, and she's a completely independent investigator at the moment. So a little bit very quick, I know you're getting tired. So one of the things that I also wanted to study is the role of glutathione in your eye. How is in the eye development? So to make a long story short and very, very quick, when we knock out the gene, especially in your eyes, the mice develop a microphthalmia. I don't have to say for the, it's small eye. All right, and this is very well defined. We have studied, we have published it. And in addition to that, what we found, which was very unique, is the eyes. What we found immune-related processes were upregulated in the lenses of these knockout mice, which brings me to the next slide, that we found immune cells infiltrate the eyes of the GCLC knockout mice, which this is unheard of because your eye is supposed to be very strong and never have an inflammatory. You can see leukocytes and macrophages in here. This project has generated really high-end publications for the immune system and everything. So, but one of the things that I am super excited. Initially, I wasn't planning to do that because we're still trying to get a patent on that, but heck, I'm getting an honorary degree in University of Athens. I think you guys deserve to hear that story, even if I lose the patent. I don't care. So what we wanted to do is we wanted to determine the role of glutathione in the pancreas. It's a major antioxidant. You want to see what's going to happen if you knock out the glutathione from the pancreatic islets. So through genetic manipulations, we were able to knock the gene out of the endocrine pancreas. The results, this is my students committee, which are established investigators in the diabetes field. They said they have never seen a phenotype like that ever. We haven't published it yet because we want to publish it in Nature. We want to publish it by knowing the mechanism. We don't know the exact mechanism yet, but the mice are perfectly fine when they're with their mother. Once they're getting out of their mother, they stop having milk from their mother. You can see that the blood glucose goes really high, sky high, and then the insulin drops completely. So you don't have insulin production. And of course, you can say, why is this happening? This is a very quick immunohistochemistry. You cannot see it right there, but essentially, the pancreatic islets disintegrate during the process after the mice have been out of their milk. This is what we have a lot of studies now. We're trying to rescue the phenotype by multiple interventions, genetic methods, and everything. But it's really important, and it's the most fascinating thing is, I want you to see here. We have it within 2 months of the life of the mice. And you're talking, this is not aged mice. This is only 2 months. It's young mice. We have a complete loss of the islets. We have complete loss of, the parts of the pancreas that they generate your glucagon and your insulin. There is no islet in there. No glutathione, no islet, and the mice essentially die from severe diabetes, most likely to type 1 diabetes, although it's not immune prevalent. So this is again both control and knockout. So as I said, we have expected and unexpected results from glutathione. This is a project that has rewarded us with some surprises, and we're going forward. So the exposome going forward, this is we have an increase, very high increase of neurodegenerative disease. We have also neurodevelopmental disease. Just the autism incidence has increased dramatically in the last 20 years. We have a lot of autistic kids. We don't know how it is. So what I have established now, and trying to generate a new center that we're studying the role of the exposome in neurodevelopmental disease. And in that case, I have developed a multi-center involving Yale, Harvard, Emory, and Aristotle universities that we are utilizing the genome exposome interactions. We're studying the genome exposome interactions by utilizing traditional and genomic epidemiology, and also utilizing novel 3D organoids, because you cannot really, if you want to use human stuff, is you going and you develop from stem cells, you're developing the 3D organoids, and you expose them to multiple effects, and you find out what's going on with other single-cell RNA metabolomics and so on. So this is something that we put our efforts in there, and but because of my duties with the P42, now I might pass this center, not the leadership of the center, to one of our opponents. So the point I want to make here, and I want to thank my dear colleague David Thompson, which really found those images for me, is I've started all my research by myself, trying to get answers, trying to do experiments, stay in University of Ioannina really late and trying to do this, and trying to do, of course, you can say that hobby is research, fishing is our profession. But essentially, I started this as a graduate student, but then you, growing up as a postdoc, you realize, hey, if we do teamwork, we can achieve much more than just doing a single one. And to that extent, if you utilize advanced technologies, which is the mass spectrometry, the artificial intelligence, the supercomputers, you can get to your destination much faster than that. Okay, so collaboration and also the new technologies can bring us to this. So they say that life is a journey, not a destination. I think science is both. I have derived enjoyment from working with creative scientists, the journey, and being involved in making discoveries, which is the destination. And I'm still so excited. David Thompson says, I have this infectious passion that I transfer to all of my trainees, which I still do, you know, and I have it. So with that, I want to thank my lab, my students. Not all of them are in there, but at least, you know, most of them are there. Among them, I think the rector mentioned that I have also a passion to train Greeks. Right now, in my team, I have Georgia Tsarkoftaki, who is actually a graduate from the School of Pharmacy here at the University of Athens, who is leading all my metabolomic facility, and I've got a number of students. This two of my colleagues, David Thompson, has been with me for more than 30 years, and Kirill Veselko from Imperial, we're doing a lot of deep learning and artificial intelligence. I've been constantly funded by NIH and its multiple, the National Institute of Environmental Health, the alcohol, and of course from the eye. We have done quite a bit of that. So before I go to my last slide, I really want to thank, one more time, I want to thank all my colleagues from Yale. I want to thank, again, David Thompson. I want to thank Nikos Sakelaridis, who is in the audience. I want to thank my good friends that are here tonight, my college mates, Philenia Sideri, Manos Elisavakis, and Dionysios Fragiskatos, which they came here tonight to share the pleasure with me. And I also want to thank Kostas Matsoukas in here, and of course, my really good buddies in Ioannina, Takis Kokalis, and Dr. Minopoulos, which is in the audience. With that, I have to tell you, I wouldn't have accomplished any of that if I didn't have, and I know Adonia Trichopoulou is giving me always hard time because I don't praise my wife enough. So Antonia, I wouldn't have accomplished anything without my wife, and you know, and this is all my kids. We have 5 kids, including, as I called on the top right, my 2 Lopodites, which are my twins. They're 14 years old. Well, with that, I want to thank you very much. And again, what a great honor. I am deeply honored, and thank you. Thank you very much.
Πέρα στις εκδηλώσεις, σας ευχαριστούμε πάρα πολύ για τη συμμετοχή σας. Thank you very much for your participation.
Μικροβίωμα εντέρου.
Το έχουμε.